So here we are at 6.6, the cytoskeleton. So scientists used to think that organelles in the eukaryotic cell were free-floating. However, they are not. They're attached to the cytoskeleton. The cytoskeleton has a complex role within the cell, and the only reason why we are able to see them now is improvements in microscopes, like we talked about earlier with the SEM and the TEM microscopes. And one quick note is that bacteria cells have protein fibers that act like a cytoskeleton, but the types of cytoskeleton that we're going to be talking about today are going to apply only to the eukaryotic cells. So here we have the eukaryotic cytoskeleton is going to use oppositional forces, sort of like a dome tent. You know, when you put the sticks together to make a tent and then you apply pressure and it pops out. That's sort of the dynamic tension that we're talking about with cytoskeleton. So these parts are highly dynamic. They're pretty impressive. And these parts can be dismantled and reassembled at a moment's notice, which is going to be really helpful for mobility and so forth, as you're going to see. It also provides anchor points for organelles. So like we said, the organelles and even enzyme complexes are not free-floating. They are attached, and they're going to be attached to the cytoskeletons. They also provide motility or movement. So what I have for you here is a video. So we're going to have a video within a video. What I want you to do is to go to the main screen or the website that you access this video and go to the description section and click on this first link. And it's going to give you about a 4-minute video about the sort of the introduction to the types of cytoskeletons. And so I want you to watch that and then come back to this. So I'm hoping that you just watched that video and we're now talking about microtubules and you have a brief understanding of what microtubules are. They are one of the 3 types of structures that are used in the cytoskeleton. They're the largest in diameter, so that they're the one that kind of has a tube shape, and they're made from a protein called tubulin. The tubulin exists in the cytosol, and it's going to, whenever there's a signal, it's going to attach onto the plus end. It's going to attach onto the end of the to the signal, and it's going to grow. So this is how it's going to assemble. Is this one side is going to grow. So they play many roles. Microtubulin plays many roles. It supports the shape of the cell. It's going to be a track for vesicles to move through to and through the Golgi apparatus, and also tracks for organelles with motor proteins. So they actually walk across these tracks, allowing for movement of different things within the cell. Cilia and flagella are types of microtubules, and they are responsible for motility. So flagella, they move with an undulating motion, sort of like if you could picture a dolphin swimming in the ocean. That's more of an undulating motion, whereas cilia have that back and forth, that power stroke where it goes back and forth, like oars rowing a boat. So they can be used, so the cilia are used to move materials along a track. So if you have saliva that needs to move down your trachea, it's going to, all of these cilia, which you can see here, are going to float and help push the materials down where they need to go. Also, fallopian tubes are going to move the eggs towards the uterus. So these can be also used, this is pretty cool, as an antennae. Most vertebrate cells contain a single primary cilium. This primary cilium is used for communication. It triggers a signal transduction pathway, which allows for communication between cells. And this is going to be very important in brain function and embryonic development. There's lots of cells signaling other cells during embryonic development. And so what I have here for you are a couple videos about microtubules, about the 9+2 pattern. So we can see that here we have the 2, and there's going to be 2 sets of 9, within these basal bodies, or 3, 9 within the basal body. But so to watch these 2 videos, so I'm going to give you time to click on this second and third video to watch to give you an understanding of the 9+2 and the 9+3 pattern and how these microtubules can help cells with motility or for movement. So next we have microfilaments. We went from the largest in diameter of the 3 types of cytoskeleton to the smallest in diameter. So I like to think of microfill. Fill is a soft eye, thin is a soft eye. So microfilaments. These are solid rods. They're made of a double twisted chain of actin. So if you have like a 1 chain of actin, you have 2 chains of actin, they're going to sort of wrap around each other and make a long chain just like that. They form branching networks. It's a cool molecule that can grow just as fast as it can disassemble, and they're present in all eukaryotic cells. So they exist just below the lipid membrane, and they create that tension, a 3D tension in the structure. And they actually, because there's so much pressure on that cytosol, it's going to create a firmer region on the outside of the cytosol, so that goo becomes more dense, and that's called a cortex of the cell. That outer region that is more firm because there's greater pressure being applied by these microfilaments. They're used in combination with myosin. So whenever you talk about muscles, you're going to talk about actin and myosin. And they extend for motility for amoeba. So the pseudopod or the fake foot of the amoeba. So these are going to be able to grow and retract because of the ability for those microfilaments to grow and disassemble very, very quickly. And for cytoplasmic streaming, which is movement of organelles within the cell. So to see a little bit more information about the microfilament, please click on the next URL for microfilaments in the discussion box on the web page. And we'll be moving on to intermediate filaments. For our last cytoskeletal element, we're talking about intermediate filaments. They are named for their diameter, so they are the intermediate. They are smaller than the microtubules, which are the big tubes, and they are larger than the microfilaments, which are the thin rods. The function of these are truly the sort of long-term load-bearing skeletal elements. They are composed of keratin, so they are structurally sound. And if you think of like an animal that goes off into the woods and dies, and a while later you're going to see the remains, the skeletal remains. This is what I sort of think of when I think of intermediate filaments, because they're going to exist even after the cell breaks apart or breaks down. They're still going to be there. They're still going to be around because they are they're made of keratin and they're very structurally sound. So they're going to give points of contact for organelles to connect to the cytoskeleton, and the nucleus is going to sit inside of a cage made of intermediate filaments, which is going to protect it and keep it in a very specific spot. So if we look here, we have intermediate filaments that are connected to the microvilli right here, and this is going to be keeping it in place and making sure that the microvilli are structurally sound and connected. It's the anchor. So this is 6.6 in intermediate filaments, and those are the elements of the cytoskeleton. Have a great day.